Here I'm going to explain the three different pathways of the complement system, or 3 different cascades they call it. The complement system, we know, is a defensive system consisting of over 30 proteins, different proteins. These are produced by the liver and found in blood serum. The complement kills microbes in 3 different ways: opsonization, inflammation, and cytolysis. So those are the 3 ways that this process gets rid of the microbes. It is called a cascade system. What the word cascade means is, one reaction triggers another reaction, and that triggers another reaction. So it's kind of a good name for it, because that's kind of what happens. It goes on and on and on and on and on. It can grow very fast, one after the other. The cascade activation, in the cascade activation, the proteins are they're designated with a big letter, like letter C, with an uppercase letter, like letter C, and are inactive until they split, which it will have a lowercase letter. So, like for example, C1. When they split and become active, it will be C1A or C1B, and that's with a lowercase letter. There are 3 different pathways in this system, in the complement system, and I have a little paper right here showing a little graph of what the difference between them. But I'm just going to briefly explain it. You have the classical pathway, the alternative pathway, and the mB-lectin pathway. And I'll start talking about the classical. The classical pathway, it relies on antibodies to initiate it. It has antigen-antibody complexes. In this pathway, C1, this is the protein in this pathway, it becomes activated when it binds to the ends of the antibodies. So, once that is activated, C2 and C4 it activates these proteins once C1 is activated, and it cuts these in half, and then C2 is cleaved into CA and B, and then C4 is into A and B. So it's kind of like you go from C1 to C2 and C4, and then C2 and C4 are split: C2A, C2B, C4, C4A, or C4B, C4A. So those are activated, and they're cleaved. It's kind of what I was talking about just a minute ago with the activation wise. The C2B and C4B, they bind together and they form what's called the C3 activation complex. These activate the C3 proteins in this pathway. And I could go in more detail about, the difference in C3A and all that. But then eventually it goes all the way down to C, where the C5 complex is created. When the C3B and binds to C2B and C4B and all that, it can get a little complicated, but eventually that's what's created. Then you have what's called the C5 activation complex, and this activates the C5 proteins, of course, and obviously that's cleaved into C5A and C5B, as all the other ones. And these are done by the C5 activation complex, and then these actually coat the surface of the bacteria. So the C5A disperses away from the bacteria, and it binds to mast cells, and it makes inflammation increase, and these are where leukocytes are found. And then, of course, you have the MAC complex here, or the membrane attack complex. the C5, basically how this is built, the C5 binds to C6, and C5 and C6 activates C6, so that can bind to C7, and C7 binds with C8, and then binds with C9. All this forms the MAC complex, or the MAC. And then this complex, what this does in classical pathway, it causes cytolysis. This is basically the attack of the or the loss of cellular contents through the channel by membrane attack. So that's what that does. And then we have the alternative pathway. With the alternative pathway, it does not need antibodies to initiate it. It's slower than the classical, and it takes place on pathogen surfaces. And I'm not going to get too in detail with that one, but with this pathway, you have C3, and it's an unstable, I don't know how to say this, diester bond. This bond makes C3 slower, and so it makes it subject to spontaneous hydrolysis to C3B and C3A. C3B can bind to foreign surfaces, and all that, and then you have something called factor B that comes into play after C3B and C3A split. And then factor B is bound to C3B. Then you have something, the binding of C3B and factor B allows the enzyme called factor D to cleave factor B to BA and BB. So that splits that up. And then we have something called the C3 activation complex here. Properdin is called factor P. So we have factor D, C, and factor P. This binds the C3 BB complex, and it stabilizes that. And then there's a few more details I go in with that one. I'm just going to stop right there. And then we have something called the MB-lectin pathway, which is the last pathway. And this one was the most recent discovered. It's called the MBO pathway or MB-lectin pathway. It can activate the complement system, and it's called, like they call it, an antibody-independent fashion. So it's independent of the antibodies. It doesn't have to. It does not depend on the antibodies to be activated. It's activated by the binding of the mannose binding lectin, or MBL, to carbohydrates on the surface of pathogens, which I stated earlier. So right here we have like a little chart right there of showing you everything in a gist. So let's get this right. So we have 3 different pathways here. We have the classical, the MB-lectin, and alternative. As you can see, the classical is antibody, relies on antibodies. It has antigen antibody complexes. The lectin binding is next. Lectin binding to pathogen surfaces, which I just stated, and then the alternative pathway binds to pathogen surfaces. Then that comes down to create the complement activation. This is where the C3 convertases takes place, and I'll explain that in just a minute in a different picture. And then you have recruitment of inflammatory cells as in product of that, opsonization of pathogens and killing of pathogens. And basically here I have the same chart. It just shows a little bit more in detail and what proteins we have taking place there. So in the classical, the antigen-antibody complexes. We have C1q, C1r, C1s, C4, C2, the MB-lectin, mannose binding lectin, which I stated a minute ago, binds mannose on the surfaces of pathogens. We got MBL, which I mentioned. You have MASP1, MASP2, C4 and C2, and then alternative, you got bind pathogen surfaces, and C3, B, and D, and then that C3 convertase, which I told you I mentioned, that binds that C3 convertase. Then the product of that is C3A and C5A, and then C3B right there in the middle. And then the terminal complement components, C5B, C6, C7, C8, C9, all that, which I stated makes that membrane attack complex in the alternative pathway. The MAC in lysis of certain pathogens and cells happens here. So those pathogens are destroyed there. Then under C3B, binds to complement structures on phagocytes. You have the opsonization happening here, which is one of the ways that we get rid of those pathogens. And then C3A and C5A, the peptide mediators of inflammation. Phagocyte recruitment happens here. So that's a good little gist of those three pathways. Explain the best way I know how. So we have the classical MB-lectin pathway and alternative pathway as far as the three cascades of the complement system.